Hello, my name is Guillaume Noguier. I am the R&D director of Teradia company, and I'm going to present you results about the development and evaluation of eye tracker Vedolizumab and eye tracker anti-Vedolizumab kits. I have nothing to declare. Vedolizumab is a monoclonal antibody directed against alpha-4 beta-7 integrin. This monoclonal antibody is used for the treatment of inflammatory diseases as Crohn's disease or ulcerative colitis. And the monitoring is already proposed with ELISA technique. iTrack10 platform is based on chemiluminescence technology. It is a fully automated random access platform. And currently, several eye tracker kits have been already developed for the therapeutic drug monitoring. For example, monitoring of infliximab, adalimumab, and ustekinumab are already available. To remember, therapeutic drug monitoring is a clinical practice of measuring serum drug and anti-drug antibodies concentrations to guide clinical decision-making. Therapeutic drug monitoring is now seen as one of the cornerstones of personalized medicine, used to improve the treatment of patients treated by biologics. In order to increase the panel of drug monitored, we wanted to develop additional assays, and especially eye tracker Vedolizumab and eye tracker anti-Vedolizumab kits. So what about the methods used to detect and quantify Vedolizumab and anti-Vedolizumab antibodies? For Vedolizumab assay, we used magnetic beads coated with an anti-idiotypic anti-Vedolizumab antibody. Serum sample is added to the beads, and if Vedolizumab is present in the samples, it binds to the coated beads during 15 minutes at 37 degrees. After a wash step, anti-Vedolizumab polyclonal antibody conjugated to acridinium ester is added during 15 minutes at 37 degrees. Finally, 2 triggers solutions are added after a wash steps, and the light emission is quantified with a photomultiplicator. The quantity of light is proportional to the concentration of Vedolizumab. For anti-Vedolizumab assay, a bridge format assay was used. Indeed, we used magnetic beads coated with Vedolizumab to capture anti-Vedolizumab antibodies, and we used Vedolizumab conjugated to acridinium ester for the detection of anti-drug antibodies. And as for Vedolizumab assay, time and temperature of incubation were the same. So let's go to the results. What about the performances of the assays? Accuracy of Vedolizumab assay was assessed in using 3 human serum samples added with different levels of Vedolizumab. Percentages of recovery, which is defined as the ratio between the drugs level detected and the drugs level added to the sample, were calculated. All percentages of recovery were compressed between 84% and 104%. The acceptance criteria of 80 to 120% were reached. So we can say that human metrics have no impact on Vedolizumab quantification. For the intra-run precision of Vedolizumab and anti-Vedolizumab assays, we used clinical samples. Clinical samples at low, mid, and high levels were tested 10 times within a run. And as you can see on the figure, all coefficients of variations, also called CVs, were below 5%. High intra-run precision is reached for both assays. For the inter-run precision, of vedolizumab and anti-vedolizumab assays. Clinical samples at low, mid, and high levels were tested on 6 independent runs. As you can see on the picture, all CVs were below 10%. So we can say that high inter-run precision is reached for both assays. During the development of the assays, the lower limit of quantification, also called LLOQ, were selected at 1 microgram per ml for vedolizumab and 10 nanogram per ml for anti-vedolizumab antibodies. The lower limit of quantification were validated in using 110 samples for untreated patients for vedolizumab assays and in using 118 samples for anti-vedolizumab assay. And as you can see, whatever the assay, all samples were found below the respective LLOQ. So we can say that both assays have high specificity at their respective LLOQ. Additional tests were performed to assess the specificity of the assays. Samples with high concentration of potential interfering agents as bilirubin, hemoglobin, rheumatoid factors, but also triglycerides and biotin, were spiked with low and high levels of vedolizumab. Same kind of tests were performed with low and high levels sample for anti-vedolizumab antibodies. The percentages of bias, which is defined as the difference between the levels of drug and without the interfering agents, were calculated. The acceptance criteria of 20% of bias were reached for both assays. Quantification of vedolizumab and anti-vedolizumab antibodies were not impacted by interfering agents. Finally, we compared eye-tracker assays to LISA tracker assay based on ELISA technology. For vedolizumab assays, 33 serum samples from treated patients were tested, and we found a coefficient of determination of 0.96. For anti-vedolizumab assays, 24 serum samples with anti-vedolizumab antibodies were tested, and we found a Spearman's coefficient of 0.84. So we can say that high correlations were found between eye tracker assays and LISA tracker assays. In conclusion, eye tracker vedolizumab and eye tracker anti-vedolizumab kits are innovative assays which exhibit fast, accurate, and reproducible results. Excellent agreements were observed with LISA tracker assays. Eye tracker kits are valuable tools for the monitoring of patients treated with vedolizumab and allowing rapid treatment adjustment. I would like to thank you for your attention, and don't hesitate to send questions at the following address. And for the final word, I would like to thank all the people involved in this project. Thank you.